I guess we can start. My name is Emma Turian, and I'm teaching at Northeastern Illinois University. I'm in the math department, and I created a course called Math 371. It's Mathematical Modeling for Cancer Risk Assessment. And this course is interdisciplinary. This is an elective for most of the programs. So basically, I have students in the class from the biology department, from psychology, from computer science, mathematics, and other departments from the school. Most of them are from biology and from mathematics and computer sciences. This talk is pretty much a review of what I'm doing in the class. So thank you for coming. This is the outline of the talk. So I'm going to discuss how we are building models from data. Models used for cancer modeling. We usually start with growth models, and we continue with reduction under therapy. So along the way, we will have examples, free resources and codes, and references. So the functions used to model tumor growth, when we cover this topic, basically we go over parameters, which ones are the parameters, the dependent variables, the independent variables. We discuss the shape of the function, units of measurement for the evolution equation and for their solution. So we also, we also analyze then the data. We try to estimate parameter values from visualizing the data first, and then we estimate parameters by using regression or other means. So usually I like to place side by side the evolution equation and the solution of the equation. So there they can see what's the difference between plotting dC to dt. In here, C is the tumor size. It can be measured as a volume or as a cell density. So this rate dC to dt is a function of C. So we see that the rate is increasing in here with C, and the solution, which is obviously exponential. So what we go over in class is, we like to do these parallels between the equation and solution and discuss them in parallel. And then we discuss the sources for how to find parameter values, because we cannot plot the functions without the parameter values. So in this case, K can be looked as ln of 2 over TP, where TP is the doubling time. So in our case, for tumors, there are books with tables specifying the doubling time for different type of tumors and different line cells. And we also need to find the initial size of the tumor, which is denoted in here by C0. We can get parameter values from experimental literature or from the lab data. Speaking of which, so if we have this data, which the source of this data is from a semiotic paper, this represents the mouse tumor volume denoted in C. I usually use C because most of the times we work with cell densities. So it's C for cells as a function of time in hours. So we discuss data in class, and what students are seeing is that the data increasing, and then there is more scattering towards the end. Now, in all cases, for this different type of ways of performing regression, then using solver and then dynamic K, we used the same initial tumor value, which is 1.041, and this is millimeter cubes. So If we use regression with, again, C0 fixed, we get this K value of 0.0943. If we use solver, and for solver we minimize the sum of square error, then we get a slightly different K value. If we use solver with a relative sum of square error, and the reason why a relative sum of square error will be appropriate in here is because the scattering is different, especially towards the end of this function. So it's appropriate in that case to use a relative sum of square error and minimize the relative sum of square error instead of sum of square error. So if that's what we use, then the K is slightly different from when we minimize the regular sum of square error. And then we also use a dynamic K. I'm calling it a dynamic K. So what it is a difference equation for the K, and I'm taking the mean. In this case, I'm taking the mean of all those K values after removing some very extreme outliers, something like 4 times the IQR on either side of the first quantile and third one. So K, in this case, it's a bit higher. So this is the highest K we get from the dynamic K. So the results are here, shown in this graph. So the data is in red. The dynamic K with the dynamic K exponential function, this is the highest growing one. And then we have the regression, and then the two solutions, the two trajectories from using K from the solver with the relative sum of square error and the solver where we minimize just the sum of square error. You see that they are a bit different. So having this, we realized that with the students that the exponential function is not the best fit for this data because the growth slows down after a while. So what we need is a model that includes a slowdown of the growth rate later on when the tumor is larger. So the new rate is going to be, we're choosing to have a rate that declines linearly with increased tumor size. So it takes this format, which is the well-known logistic function. Now we look at different growth functions, and we also look at some of the functions which are derived from first principles, so not just empirical functions, but the logistic functions is loved by mathematicians because, for example, in comparison with the Gamperts, it doesn't have the log. So it's easier to work with the logistic growth. However, the logistic growth has an inflection point at the m/2. So I'm using m for the carrying capacity. At m/2, there is an inflection point, and compared to the other functions, such as Gamperts, for instance, the logistic bends quite a bit around the inflection point, which makes it unpractical for doing data fitting because it's very unlikely that the data bends the exactly same way as the logistic function. So again, we look in parallel at the evolution equation and at the solution. So basically, as the rate increases, the function grows faster, the rate decreases, the function slows down after the inflection point. Okay, so again, the C0 is before m/2, before the inflection point in this case. So moving forward, we're trying to fit this model, the logistic model, to the data and to use, again, regression, the solver where we minimize the sum of square error, and the solver where we minimize the relative sum of square error. And again, we take the mean of this, what I'm calling it, the dynamic K. So the dynamic K is the highest among all the K values that were obtained in here. And the solution looks like this. So obviously, it's okay to have something like the logistic equation when using the dynamic K value, which is not a good fit. And we discuss it with the students as to why this is not a good fit. So they need to explain why this dynamic K value does not give us a good fit for our data. So obviously, because of so many bendings of the logistic function. So next, I like to dabble in the somewhere in the field of machine learning. So we use the random forest algorithm, and we take the best fit data from these logistic functions in here, and we build normal distributions somewhere inside within each sub-interval, and with the mean being the exactly value of the function. And we create a training data set from sampling from a normal distribution. And then we compute the mean absolute error. And as you can see in here, there isn't a better fit for the random forest algorithm versus the logistic, the previous green line. We also use the random forest using randomly generated numbers from a uniform distribution this time, from within each subinterval. So in other words, we're assuming that the oncologist or the researcher would know the values from within each subinterval, with the exception of the actual data points, which we use them for testing. So, so this way, students get to see the results from testing the model using the original data values. So in either case, the mean absolute error is not that much different. Also, if we split the actual data set, which is not large, we have only about 24 data points. So if we split that into, let's say, 87.5% and 12.5%, which is the train and the test, and we're testing on these 3 points in here, this is what we obtain. So again, because of the large scattering towards the end, it's very hard to fit the data well when the tumor is large. Now that we know that the tumor is growing, we need to use some therapy to treat the tumor. And the way we present this immunotherapy approach is to first start with an assumption for tumor growth. So I'm staying in here with the same tumor growth, which is logistic. And we start with the logistic growth, and then we use the diagram to add more terms to this system. So we look at the diagram and locate C. C appears in 3 spots in here, in this bubble, actually in 2 spots, but there are 3 arrows that go towards C. So no, two of them going towards C and 1 of them away from C, and we add the terms according to the arrows going towards or from the C. So terms are added to the system according to this diagram, as is the complexing. So what's happening in here is the effector cells, which are the immune cells, are starting to fight with the cancer cells, and they create a complex, and there are 3 things that are happening from this complex. So from this complex, they can go back to the way they were, the cancer cells and the effector cells, or The cancer cells are dying, or the effector cells are not really dying, but they become inactive. So we populate these equations with the respective terms, and we end up with something like this, where C total is the number of cells in the tumor plus the complex. So moving on, we add more assumptions to the system, and I like to draw this parallel between the assumption and the equation. So I'm placing the, for instance, A, the assumption A goes to this term where A is seen in here. So that represents a constant rate of effector cells denoted by alpha. Effector cells are clearing, so they're dying and clearing according to this term in here. And we also have all the other terms for the death of the effector cells and the cancer cells. And then we have a very interesting term in here, which is the assumption E, and that's for the recruitment of the effector cells to the tumor site, and that's the Michaela-Menton term, MM. So now, based on our experimental observation, the system is being reduced to these two equations in here. And we are plotting the equations, the number of cells as functions of time, which they are being seen in the plot on the left. So the cancer cells are in red and the effector cells are in green. And we also have this CE plane in here with the time. So these were done in R. Okay, so also what we can do with mosaic calc package in R is to create a streamline field and a flow field. I found this very interesting and very visual for the students and very easy to use. I also included the code in here. In here, I and double I are the right-hand side. So these are the I and double I. The right-hand side of the two equations from a system of equations 2. Okay, so also what we do is compare the different trajectories. And again, I included the code in here for the two trajectories. So what's good about mosaic calc is that it provides the time. So the labels for the two trajectories, T1 and T2, the labels, again, they represent time at which those points are. So the mark times are a bonus. And I hope you find the code useful. Then we discuss, once we have these trajectories, we discuss actual values. What do they mean? The trajectory shown on the left is identical to T1 or T2. And what does the vector plot on the right suggest? Is tumor dynamic sensitive to the initial tumor size? Is the dynamic sensitive to the number of effector cells? So we get to discuss these two things in here. Now, another free software is MATLAB Plotter, written by John Polking of Rice University. So the only issue I've seen in here is that we need to dabble with these settings, and these are the precise settings that I used to obtain these plots. So the arrow length had to be changed because initially with the default arrow length and the number of arrows, I couldn't see anything in the plot. So I had to make changes to these settings that are in here, so I can end up with this vector plot that we are seeing in here. Okay. So a two-population model can be non-dimensionalized, and the Kuznetsov-Taylor system is seen in equation 3, and we have the null lines. and we have up to 4 possible steady states. Now, what we would like to see is a tumor-free equilibrium, which is in this case being given by this point in here, sigma over delta and 0. And obviously this is coming from the intersection of two null lines. Now, the stability of this point depends on parameter values. So what we're seeing in here is the Jacobian, and based on the Jacobian, there is a theorem which determines the stability based on the determinant of J and the trace of J, the sign of the determinant of J and the trace. And this is an example of a stable dormant tumor point. And it's a dormant tumor with obviously a relatively low number of tumor cells. Okay. So now going through a 3-population model. Now this is not a classical 3-population model, but then again, the mosaic calc is helpful because we can draw a trajectory, and I included a code underneath in here. If we hover above the trajectory, we get the coordinates, which is very, very helpful because sometimes it's difficult to visualize a 3-dimensional system and get to see when is it that the effector cells are going down, and then the T, which are the T cells going up, what's happening with the tumor size. So it's very good to have this hovering property that provides the XYZ coordinates. And this is just the picture that we're seeing on the right side. These are the references, and thank you for listening. I'm gonna stop sharing, and let's see if you have any questions, if you like me to go back to anything.
That was a very nice presentation, Emma. Thank you.
Thank
I
you,
want
Ben.
to go through it more slowly when we're not online, but
yeah, I do have the lecture book on the internet as a preprint, and I have more codes in there, and I have quizzes and projects and exercises. So the lecture book has exercises and case studies within each, not quite within each, but somewhere inside the lecture slides. So as we cover material, we stop for questions, for exercises, and for case studies. So if you want, I can put it in the chat if
yes,
anyone,
that would be lovely.
If anyone is interested in taking a look at this. Okay, so where is the chat? Okay, so I'm finding the chat. Okay, so I'm gonna type the link to the book. So, okay. In the meantime, if you have questions, other questions, I'll be happy to answer them. Okay, please let me know if you cannot access this, because it requires a login, but I'm hoping it's accessible without the login.
Okay. I've dealt with the tumor growth models back when I was teaching. I'm retired now, but I haven't thought about the immunotherapy and the fact that you can make that accessible. That's wonderful.
Yes, and there are 2 big names, Kuznetsov and Dipailas and Radanskaya.
Yes.
Yeah, they started these two population models, and they've analyzed them in very, very great detail. And then there are 3 populations and probably 10, 12 population models now. But we start with a 2 population model, obviously. And the growth, we start with the easiest one, which is the exponential growth. And we go from there. We analyze as to we look at things as to why the exponential growth is not appropriate, because the data suggests that there are limited resources, and therefore there is a flattening later on. The tumor is approaching its carrying capacity, so to speak. And then there are different approaches for the therapy. So the therapy that I'm discussing are the therapies are immunotherapy, chemotherapy, and radiation therapy. And I also have the PK/PD models, so pharmacokinetic and pharmacodynamic models. Now, for the radiation therapy, there are different approaches. Basically, each therapy has its own approach. So for the radiation therapy, we have to use a completely different approach. It's a point-to-point modeling. And that's because the dose, the radiation dose, is being given in fractions. And there is a surviving fraction that needs to be applied after each radiation fraction. So therefore, the model changes after each fraction of radiation. So I do have this in the book. I'm thinking you're able to open that.
I got your link. So I will try that another time. I can't do more than one thing at a time.
Oh, that's okay. But I'm hoping it's accessible and you guys don't have to log in.
Okay, thank you.
Yeah.
Question.
Yes. Yes, sir.
Yeah, my question is, so what, when you identify your model from clinical data, I assume that's what you're doing to get your parameters. What sort of clinical data do you're collecting a time series of clinical data for your, what's the clinical data that you're collecting? Is it the cancer, the tumor cell number, or how do you?
Whatever I could find, Sammy. So basically it's from the literature, because I don't have a lab. I'm now in contact with some people that who have data from the lab, but this particular data, this one in here, is from a paper, a semiotic paper, which I can show it to you at the end. It's by I believe a student. His name is Wang. Yeah, this 4, Wang. This is the student version, modeling cancer growth with differential equations.
Okay, so is it like an assay, a data from like an immunoassay, or is it image data, or that gets the?
It's coming in a table. It's from I believe a lab experiment in China. So they published this data, and I used that, I plotted, and then we discuss it with the students, and we feed different growth functions to it. We discuss the fits, which one, the goodness of the fits, and yeah. But there are so many things. I mean, the difference between minimizing the sum of square error and minimizing the relative sum of square error. So these are details that they look into, and it's interesting for them to see why why is this working versus why that's not working. Even if it's something that's not working properly in here, they need to understand why it's not working so that they use it properly next time, for when they work on a real project.
Let's see.
Hopefully.
Okay. Well, I have to leave now, but thank you very much. And I will be in touch after I look at the lecture notes. Thank you.
Yeah, please let me know, especially if you see anything that's out of place.
Okay,
I would
thank
appreciate
you.
your input. I appreciate any input, I appreciate you being here. Thank you very much.
Okay.
Thank you. That was an interesting talk.
Sammy, I like the wiki modeling, and I'm hoping my only fear about the wiki modeling is that the students will go in and mess up the public models.
No, they can't. They can't because
They can't.
you can't overwrite. If you save it, it'll be saved under your name. So you'll
Okay.
have your own copy of it. So yeah.
Okay, so they can't change the existing public ones.
That's
Okay.
correct. That's correct. That is correct.
Okay.
Yeah. Do you
'Cause
have
that's-
the published parameter, like values for the parameters, or is that from the publication list that you shared?
Okay, so let me go back to it. The published ones, oh, I had the paper someplace. You mean in the paper by Wang, correct?
Yeah, or any of the models that you shared. How did you you fit some functions, and so what were the end the parameters, the different numerical parameters for the model that gave you these results? Like what is the the C value and the K value?
Okay, so C is the
Oh, I see the K values.
dependent.
You've got them right here. Okay.
Yeah, C
Oh,
is the
C is
dependent
your variable.
time.
Okay. So you
Yeah.
only have one parameter for this model then?
No, I also have M, which is the
M.
carrying capacity, but I'm
Okay.
very interested in changing in changes in K by using different methods.
Yeah, yeah. Oh, this is the equation that we typed in together. I get it now.
Yes, yes.
Yes, yes. Okay, cool.
Yeah, that's the logic. This is the one with the logistic.
Right. Okay.
Yeah.
Oh, cool. All right, cool. Neat. I'll take a closer look at it when the video comes up.
Yes, please, and let me know again. I would appreciate any feedback at all about the video, about the lecture book. So the lecture book is about it's this one. It's about 330 pages, and it has lectures with case studies. Also, we start with LaTeX. So I
Yeah.
get students into LaTeX because if they ever want to be scientists, they need to record everything in LaTeX,
Right.
because they can easily come up with a project, a poster, and I give them the end. They have templates for posters for what's that, Beamer, which is the presentation, and for an article. And then I have the lecture slides, I have quizzes, information, statistical, goodness of fits, methods of comparing the different models, and then about free software and about, what's that? I have the solutions with the codes
Mhm.
for selected case studies. Yeah, information about, I would like to keep all things in one place. It's good because this is the first time we had this class in the fall, and this is the first time I had a book, which is a huge relief compared to the other times when I was teaching this class and not have a book. It's so difficult because
Yeah.
I was posting different things on D2L, different files, and then the students would not know, they kept on asking me, where did you post this? Where did you
Yeah.
post that?
Yeah, no, it helps to have it even
Oh.
before the class begins. You've got the whole material ahead of time.
The whole material, because there is no textbook for such a class.
Yeah. I'm not in academia, but it is an interesting topic. I do have, friends and relatives that are, oncologists, and I wonder if this would be something that would be interesting to them. So I'll reach out
Well,
to.
my husband is one of them, so
Okay. Oh, well,
that's the reason why I know how to model radiation.
Okay. Well, maybe you can get some real data from him.
Yeah, but the legislation in this country makes it so complicated. So basically, someone will have to work full time to read the two-dimensional slides from the CTs and reconstruct the volume from the 2Ds on each slide.
Okay.
So it's a full-time job, and then
Yeah.
the legislation of, making the data available, it's so complicated,
Yeah.
you know.
Yeah. So it is image data. That's how you get your number of cancer cells from your images.
This particular one, it's the volume. I have no idea how they measure the volume, but the ones for radiation therapy, usually they are GTVs, so gross tumor volumes from the from the images. They reconstruct the volume from the 2Ds.
I see. Oh, cool. Yeah, that is a lot. That's a CT then, is it?
Yeah,
Yeah.
from a CT, right.
Interesting. Okay. Oh, very neat. Cool. So is your class a math class or a science class?
It's a math class. It is registered in the math department, but it's more like an interdisciplinary class because I'm including lots of information about tumors as well. And the biology students are very excited and they want to see how do we use those information, how do we use the data. So they're interested in the math, but there is a lot of biological content as well.
Right.
So yes, it's a full class. I get students from other universities coming to take it. So the provost was so excited that students from other universities are interested in this class that they got free tuition.
Oh,
So
that's great.
just as an introductory.
Yeah, well, that's great. That's great. Hopefully it'll keep going and growing. So yeah, that's great. I think applied math, where you have real kind of world examples with real kind of
Exactly.
implications is a lot more interesting. I think my first differential equations class as a student, it wasn't like that, and it wasn't interesting to me. I was kind of, I didn't see the point until I used it in the real world. So it's great that you're getting the students in, showing them the kind of real world applications in the class itself.
Yeah, they do enjoy it. They're very active. This is the most active class I've ever had to teach.
Yeah.
I mean, really, students are sorted. So I was teaching this class when the pandemic started in 2020.
Ah,
And it was such a noisy class. It was, again, a full class. And they were asking me so many questions. I had to bring someone from the biology department who was
Oh.
teaching cancer cell biology.
Mhm.
Because I say, I don't know the answer to this question. I don't know the answer to that question.
Yeah.
So I'll give you someone who's an expert on this.
Yeah.
So it was, and then they drove her crazy with questions. And I felt ashamed. I say, probably, maybe she might hate me by now, probably she would say, I did this on purpose or something. But they were so active, so crazy. And all of a sudden, the show stopped because the pandemic shut us off. So it was like from all that craziness, from
Oh,
all
that
that
was,
noise,
you mean that was in class, in person?
it was in person, yeah. And
Yeah.
then all of a sudden, nothing.
Yeah. Well, you didn't shift to remote? You didn't shift?
We did. We went to remote, but they were like
They weren't
quiet
as active.
and non-active at all. It was so flat. It was me talking.
Yeah. Interesting how things change. Things are different in person. Yeah. Different
Yeah.
dynamic. Cool. Well, I'm going to see what's on the next. I probably won't stay for very, very long. I actually have a Super Bowl party that I'm hosting, so I gotta
Oh, wow.
stay for the end of it. Anyway, I hope you enjoy the rest of the conference, and I'll take a look at your
Thank you.
video when it becomes available.
Thank you very much, Sammy.
All right.
Thank you.
Take care.
You too.
Dr. Turian?
Yes, hi, Farhan.
It was interesting. So I liked it. Actually,
But
I
please
was
call me Emma. Please call me Emma.
okay, sure. I was very interested in how you made it so ground level so that students can understand. I downloaded your lecture notes last night, and I looked through. I am interested in PK/PD models. And
Okay.
what I do in my research, I am working on fractional order differential equations. So
Okay.
orders are not one or two. In your presentation, you had one there because you are considering logistic converts or first order equations. But the equations that I am interested in, any order between 0 and 1.
Okay.
Correction. So what I am trying to do is, in the literature, mathematicians are considering Gombers. Let's say you talked about mainly logistic today. So let's say your logistic equation, when we fractionalize it, then the order becomes fractional. And they continue from there, you know, to talk about parameter estimations and other things.
Mhm.
But my goal is to look at from the ground level, like in your lecture notes, and considering reformulation of the models, like the models in fractional, not fractionalizing, but maybe, you know, something else, like fractions are there, but in another form. So that's my
Yeah.
interest. I am hoping that your lecture notes is gonna help me.
So
to do this.
basically, yeah.
And I'm a mathematician as you are. So I collaborate with medical doctors, and I have a collaborator from Basel, Switzerland. He works at the Children's Hospital, so he helps me a lot because he has the very good knowledge in mathematics and he has very good knowledge in medical science. So
Well, we should keep in touch, Ferhan. So you know my email address. I'm
yes,
gonna-
I know your email. I was really looking for someone who does this in the, you know, in the basic level so that I can ask questions when I do this for fractional. You know, the difference between the models that you are talking, they are all local. You know, derivatives are local.
Mhm.
Like first order equations are based on the derivative, first order derivative, and first order derivatives are instantaneous rate of change. But fractionals are non-local.
Mhm.
When we do fractionals, we consider, for example, tumor growth from the beginning to the time. And this includes in the derivative. That's totally different than, you know, instantaneous rate of change. So I'm hoping that, you know, this could be improved the existing, you know, models, but my plan is to start from the ground. I mean, from the undergraduate level, and then continue to get to the level that I can talk to, you know,
Yeah.
doctors.
Yeah, that's what I found. Actually, there are three, four things that I believe they're important. So starting from the ground, visualization, putting things side by side and comparing them, like the evolution equation versus the solution, because sometimes they get so confused, like which one is what are we putting, what the line that you are showing, which one is it? It's the
Yeah.
so
And
so
the thing that you said was very interesting. I have never read it before, you know, having the logistic solution inflection point there. I have never seen it or noticed it before.
yeah,
Those are the things important when we model, you know, tomorrow.
Yeah, it's one of the least practical function when it comes to fitting data, because of its bending around the inflection point, because it's very unlikely that the data will bend the exactly same way as the logistic does. So that's why oncologists usually use Gamperts.
Yeah, yeah, that's also what I did. I'm sorry, my battery is going down. Let me get my charge. Yeah, I also use Gompertz. I fractionalized the Gompertz equation. And
Yeah.
there was one medical doctor in Nashville. I live in Bowling Green, Kentucky, which is 1 hour distance from Nashville. So, so I work with a medical doctor to get her ideas about what I am doing. So I fractionalized the Gompertz, but I wanna focus on PK/PD more because this is what I am doing recently with the doctor in Basel in Switzerland.
There is a book for PKPD models. Do you?
Data analysis.
No, there is a book. Let me see.
I have a very thick book, PKPD, very thick, you know, this thick, orange color. Is that the one?
I don't think it is. Let me just go get it. Yeah, let me.
Sure.
Okay, so here is the book. Is this the one?
No.
That's a very good book. So this
Okay.
this might be very useful to you. It's by Shot. I'll put it in the chat.
Sure, you know, I need to get the,
It's the 7th edition. It's called Applied Biopharma
biopharm.
Bio. Oh, these names are,
Okay, so the author name is Sharma. Okay,
yeah.
I will search for it. I don't have that book, so I have never seen that book.
Hmm.
I usually read the papers in this area.
Yeah, so what I have is the 7th edition. It's a McGraw-Hill education book.
Okay. Okay, I took a note here. Okay, so thanks so much.
Yeah.
I will keep in touch with you because, as I said, you know,
Do you have your email? Could you put it in the chat? I can let you know if I find something interesting, you know, because I'm searching all the time for materials, and if I get anything that might interest you, or, you know, we can.
let's see.
Okay, so I'll pick it up from here.
So this is something that I really like to. So your husband is a medical doctor, or is it?
He's a physicist, radiation oncologist. Yeah, he's the chief physicist here in Chicago, in the hospital in Chicago.
My son is doing his residency in radiation oncology. So
Oh.
yeah, I'm also gonna, you know, collaborate with him to get his ideas.
Oh, it's good to have some inside information, you know.
Yeah, that's what I'm hoping, but who knows? I don't know.
They usually say, I don't have time for this.
Yeah, they say math. I don't know.
Yeah, math. Yeah, it's not really applicable.
But we can understand each other. Very nice to meeting you, and
Very nice
enjoyed
meeting
your
you.
talk.
Caroline.
Thanks so much.
Thank you.
Have a good rest of your Sunday.
You too. You too,
Okay.
Farhan.